everyone. It's nice to see you all here. Welcome and thanks for joining us to this special Variant Scientist Network event. Our first of its kind. It's called My Most Puzzling Variant. And so because it's the first of this type of event that we're doing, we'd appreciate it if you fill out the feedback form afterwards so that we can improve for our next puzzling variant event that we'll have at another later date. So today we have planned to have 4 presenters who are going to be talking about their most puzzling variants. So I know Alexa is here. Alexa Dixon will be going first. And basically what we're going to do here is everyone has about 10 minutes to present. And during that time, the presenter will be giving about 2 minutes to the audience to be able to assess the variant, see what you think about it. So let's take it away. We'll let Alexa go first. We're very interested to hear about your most puzzling variant.
Thank you so much, Yasmin. I'm going to share my screen so I can tell you about my most puzzling variant. My name is Alexa Dixon. I'm a senior clinical variant scientist at Washington University in St. Louis. The case that I have for you today is a 5-year-old female. She has mixed hearing loss, developmental delay, intellectual disability. Her mother has PCOS and endometriosis. Her father has glaucoma, repeated retinal detachment, hearing loss as an adult, and short stature. His brother, so the child's paternal uncle, also has nystagmus. This child's older sister recently failed a hearing screen in school. And so this sample, the trio, the mother, the father, and the proband, the 5-year-old female, were sent for exome sequencing at Washington University in St. Louis. We did have some previous genetic testing that was done. There was a previous hearing loss panel that had been performed. They had detected 3 reportable variants, MYO7A pathogenic variant. That gene is associated with autosomal recessive hearing loss, so there was only 1 pathogenic variant detected, so a carrier status. There was also a VUS in MYH7B, as well as in WFS1, associated with Wolfram syndrome. On exome sequencing, however, we detected a TBX2 frame shift variant. It was paternally inherited, and so this turns out to be my most puzzling variant. And I'm going to start a 2-minute timer now and have the audience try to answer the question, is this reportable? You have about 40 more seconds. Okay, time is up. If anybody, that's exactly what I was about to ask people to do, Ronak, is to put anything that they found in the chat. So, spoiler alert, there is a publication that we found that was significant, and Ronak has put it in the chat. Here's some screenshots from this publication. So, there was a potential for a novel gene disease association here. So these heterozygous TBX frameshift variants cause a novel syndrome with hearing loss and incompletely penetrant nystagmus. And I don't have the time to go through the entire publication today, but suffice it to say, there was a significant amount of family segregation data that you can see on the screen now. There were some functional data as well. They also created a mouse model, and that recapitulated the hearing.
Oh, it looks like we lost Alexa. So what I'll do is I'll look at the chat in the meantime, because I see quite a few comments here. Okay. Oh, actually, no. So I see that the paper has been posted into the chat. And then it says, I was going to say yes, based on the description of TBX2 gene function. Okay, can you say a little bit more on the function of TBX2, the protein? Nonsense mediated decay, expected loss of function as a likely mechanism. True? Well, that's unfortunate. We've lost Alexa at exactly the most exciting moment. We'll give her a minute to come back. And if not, then what we'll do is we'll move on to the next speaker, and hopefully Alexa can come back in time to finish her talk. So let's just give her a minute. Sorry about this, folks.
Apologies for the technical difficulties. Can you hear me?
Yes. Yes.
Sorry about that. I'll wrap it up quickly so that we can have enough time for everyone else. Hopefully you can see my screen again.
Oh, no worries. You don't need to rush. You came back very quickly.
Okay, great. Can you see my screen again?
Yes.
Okay. So again, mouse model, recapitulating hearing loss. If you go to the link that was put in the chat, there's also a video of the mice having the nystagmus as well. So recapitulating the phenotype of the families that were included. There was multiple families, not just the large pedigree I showed. So at our institution, we use the ClinGen gene disease validity matrix in order to establish novel gene disease associations like this. I don't have the time to go through all of it today, but this is a brief snapshot of the data that I collected. So in the publication, there were two families that both had loss of function frameshift variants. So I scored the 1.5 points for each of those variants. And then there was some functional data, so I upgraded each variant by that half a point. So that was 2 points each, a total of 4. Then again, we had multiple families. One was quite large, and the variant in that family segregated with disease, the variant and the variants both segregated with disease in their respective families. So I scored 3 points for that. There was also some additional experimental evidence that I scored as well with 2 points. And so 7 plus 2, I got a total of 9 points, which leads to a moderate gene disease association. So Ronak was able to find the gene disease association rather quickly in the chat, but this is how we found the novel gene disease association. Our laboratory uses the Franklin by Genoox software, and on the publications tab, you can see here at the top, the second publication down had this novel gene disease association. Here's a brief snapshot of the two variants that were detected in the publication. As a reminder, our variant is C.502_515 del. And so it's upstream of the two previously detected variants, but still would cause a frameshift and theoretically nonsense mediated decay. And so how would we classify this variant? I think the first thing to remember is that for a moderate gene disease association, the classification, at least for our institution, would be limited to likely pathogenic. As you can see here, for definitive and strong, you can get all the way up to pathogenic, but for moderate gene disease associations, likely pathogenic is the highest classification level that we allow. So with that, the variant was classified as likely pathogenic with the PVS1. They did show that the nonsense mediated, they did show that the loss of function was the disease mechanism. So we scored PVS1 and then we scored PM2 for rarity in the population and came up with a likely pathogenic variant. However, there's a question, is this the diagnosis for the patient? I think it clearly explains the hearing loss. However, there's also developmental delay and intellectual disability that were described in our patient. There was no mention of those phenotypes in the publication. So it's not clear if this TBX2 would be associated with that particular phenotype. So we do have at least a partial description. I think there's also the possibility for familial testing that could occur. We know the father carries the variant, but we also know she has a paternal uncle that has a similar kind of phenotype. And so I think it would be worthwhile to test that paternal uncle as well as potentially testing the sister who recently failed her hearing test at school. We know the nystagmus is incompletely penetrant, so it's possible that she also carries the variant. So in conclusion, we have a novel gene disease association that we reported in this patient. The variant was classified as likely pathogenic, which gave at least a partial explanation for the phenotype. I do wonder about the possibility of just not enough families or potential variants being detected, that potentially it could be associated with developmental delay or intellectual disability. This gene is a transcription factor that's expressed throughout development, so it's possible there's additional phenotypes out there waiting to be described, and we did recommend familial testing. And that's all I have. Yasmin.
Great. Thank you, Alexa. That was great. Just a quick question. At what age was the mixed hearing loss diagnosed in the 5-year-old girl in your program?
That's a great question. I don't remember the answer to that. Yeah, I think it was young.
Yeah.
I think it was relatively young. I don't recall it being diagnosed at birth.
And I know there's a lot of comments, but I would like to pick one just to save time. So Jenny Goldstein asked, would it be worth contacting Matchmaker Exchange to look for other cases?
I think that's an excellent suggestion, Jenny. I think that I should definitely do that. I have not yet done that. But yeah, it would be great to be able to publish some additional cases. And there's other comments in the chat about submitting that to the GenCC for community use. I think that's definitely something that I should do. So great suggestions in the chat.
Great. Thank you so much, Alexa. That was a lot of fun. Thanks for giving us the time to look over the variant. Okay, next up we have Giovanna Torresan.
Hello, Yasmin. Can you hear me well?
Yes, we can hear you.
Okay. So let's see my screen. Can you now see my presentation?
Yes.
Okay. So thank you guys for the organizing such a nice group and such nice meetings. I selected as my most challenging variant a variant in PVS2 gene that I will show in the couple of next slides. So the patient that was the index case for this variant was a young male patient with colorectal cancer at age 25. We didn't know any of his family history of cancer because this patient is adopted. And for colorectal cancer is standard to perform immunohistochemistry of the mismatch repair proteins. And this patient was identified as MLH1 PMS2 deficient tumor, indicating mismatch repair deficiency in the tumor. And this led to the clinical suspicion of Lynch syndrome. So in 2020, the patient performed a germline hereditary cancer panel, and the result of the panel was the detection of a variant in PMS2 gene. Is a frame shift variant located in exon 13. The gene has 15 exons, and one information here was the variant was described as heterozygous, but with a variant allele frequency of 20%. So here now is when you do your a little bit of search for 2 minutes. Here's the a QR code for the ClinVar if you want to check the variant in ClinVar. And what do you think would be the codes and or questions that we will need to ask for this case? I'm gonna set the timer. You have more 30 seconds.
Giovanna, this is germline
Yep.
testing, right? So in the blood?
Yeah, sorry, this is germline testing.
Yeah.
Blood.
Perfect. Perfect. Thank you.
Okay, so let's get into the context of PMS2 for people that don't work much with this gene. As we saw in the germline testing, we saw allele frequency that's lower than expected for a germline variant. It was in 20% of the blood. And PMS2 is a gene that's very technically challenging to sequence with short reads and EGS because it has multiple pseudogenes. The most important one is this pseudogene called PMS2CL, where all exons from 11 to 15 and also exon 9 are very similar, homologous to the PMS2 gene, so sequences are very equal. And our variant is here located at exon 13. So this is the view that we had of the variant at IGV and also the view of the variant in our secondary analysis software. So we see the variant here, the allele frequency, and we also already see in the software this flag here that indicates that this is a pseudogene region that can have problems with the homology. And when we looked at the IGV, I saw that this variant actually, we already seen this variant before, but not with this nomenclature. So annotated like this, it was the first time it appeared at our database, but looking back at older cases, we saw that this variant was actually already detected in other cases with a different nomenclature from the software. So the variant now was considered indel, so insert deletion with an insertion, but we had prior patients detected with this deletion of two nucleotides combined also with a missense variant that was this T2C change here. So we went back also to all these cases to look at the frequency of this variant in both nomenclatures at our database. We had 647 patients at the time we end up to do this analysis for the paper that we published. And we saw that the variant was detected in 1.8% of our cohort. Until 2019, the variant was called as this deletion, and then since 20, the annotation software called this as a Darwin's variant. And here's our probe of index case here that was the first where the nomenclature had a change. So this was a lot of red flags for us because this first variant we already had validated that the variant is actually in the pseudogene and not of the gene. So we did the same to show that the variant was not located in PMS2 gene. When we looked for this, the second variant, the second annotation, if we look at it in NOMAD or other populational database, there is no description of this variant. It's like it doesn't exist. But if we looked at the deletion, the first annotation, we can see that this variant has a frequency of 2.5 to up to 5% in the African population. So this is another indication that this variant actually is not happening in the gene, because this is way higher than the incidence of Lynch syndrome that we would expect for any population. We did other orthogonal tests to prove that the variant was actually in the pseudogene. So we did RNA-seq for 2 patients, and we also developed gene-specific PCR that uses intronic mismatches between the 2 genes to be able to amplify these genes, the gene and the pseudogene separately. So as a conclusion for this case, we could show that the variant is actually at the pseudogene and should not even be reported in the clinical report for this test. We continue to investigate the patient to understand what was the cause of this loss of expression for MLH1 and PMS2. We did a somatic comprehensive genomic panel in the tumor of the patient, and we identify a biallelic MLH1 loss of function mutation, so confirming this as a sporadic case that we used to call Lynch-like syndrome, but today we don't, just call it sporadic MLH1 mutated tumors. And as key lessons from this case, we need to remember that genes that have pseudogenes sometimes require orthogonal confirmation methods to really prove where the variant is located, and that also difference between changes and update in our secondary analysis softwares and annotation algorithms can have different nomenclatures for the same complex variants. And finally, the most important part is that population diversity in database matters, because this is the only way to classify variants correctly across different ancestries. So I'd just like to thank Anthony here, that was the PhD student involved in gathering all this data for the publications, and leave with you the QR codes for where you can find the versions of the paper. Thank you.
Thank you so much. What a wonderful presentation. What a nice story.
Yes, now it's nice.
Yes.
When it was happening, it was like kind of desperate trying to understand, but now it's really a nice story.
Yeah. And I love that the audience was already on to the pseudogene story. And I also wanted to comment on the fact that, you know, when IGV calls things, it's so important with these dalliances and, you know, these frameshifts to be able to get the right nomenclature. It can be very misleading.
Yes.
And so that's why it's so important to check the literature too, to make sure.
Yeah. And inspect the alignments, also very important for this difficult variants. Yeah.
Yeah. Interesting that he has a sporadic cancer at 25 years old. Wow.
Also.
Yeah.
Striking the age.
Well, yeah. Thank you so much. I hope you stick around to see the other 2 presentations.
I'll be here.
Yeah. Okay, great.
Thank you.
Okay, next up, no problem. Next up, we have Aksa Alam.
Can you hear me? Oh, can you hear me?
Yes, I can hear you. Yes.
Great,
Hi
great.
there.
Hi, sorry, I feel lost, like I stumbled upon all of the germline variant scientists. So just a brief introduction, I'm a somatic variant, I don't want to say somatic variant scientist, I'm a bioinformatician who analyzes the output of our clinical pipeline. And so the challenging part of the variant I'm going to present is more so on the like technical side of what we should put into the clinical report for the geneticists and other important people like the genetic counselors to do. So let me share my screen first. Share my screen. Okay. Can you guys see my screen?
Yes.
Great. Alrighty, so here is a somatic tumor contribution to this series. It maybe won't be as exciting as everybody else's, and it'll be quite short, but we'll see. So just a brief context, I'm sure a lot of people here know, but like, we look at de-identified patient data, so I don't have access to any patient information. So things like phenotype, family history, etc., I do not have access to. And then I also am not responsible for guiding clinical decisions or anything. I just finding and presenting the variants. So that's the context of this today. And so we present a clinical report, and in the clinical report, we also suggest treatment options, but these come directly from the website OncoKB, which shows us somatic variants. And if a somatic variant is oncogenic in OncoKB, like classified by OncoKB, we don't use ClinVar or anything like that yet, because I know that the somatic portion is newer. And actually, OncoKB recently has started adding germline variants too. But regardless, if it's in OncoKB and oncogenic, we report it on the report, and if it's not, we do not report it. So the case here, the bare minimum, what I can say is it's a whole genome transcriptome. In an ovarian case, this was the site of biopsy, and then there's some other statistics about the tumor, bioinformatics statistics, like the purity was relatively high and the ploidy was about 3. And so when I opened the clinical report as the interpreter, I saw this variant in BRCA2 in the tumor, somatic variant. Nonsense mutation, it had loss of heterozygosity. We all know what a nonsense mutation is, and of course it's oncogenic here. It's a truncating mutation according to OncoKB. So this is associated with some FDA-approved therapies, and so in general, good news that like there's something actionable in the report. So we go to open the math file and we see the file that contains the variants, and we see BRCA2 at this chromosome position. It's a nonsense mutation and it's a stop gain. And so if we were simply machines that took the output and spit it out to the genetic counselors, then this is what I would be reporting. But we're not machines. We go actually look at things in IGV and we look at the data. And so we saw it said a truncating mutation. I also went and opened the VCF file, and you can see some information here, which I'll come back to in a second. It's just a lot of numbers. And so you go to open IGV and you see this. And so where it's centered here is the call, the nonsense call, right? You can see that the call is TAG, right? And so that's what was reported, but there is something beside it. So I'm going to leave it here as to what you think we should do. Like, you are me, you open the IGV, you saw this right next to this variant, and you're debating whether you should report it or not. What would you do? I'll leave it to you guys to think about. Just in the last minute or so, I'm going to let anybody who knows what the VCF file, I'm sure we all know what VCF are, but to look at this data and see if they can make any conclusions off of it. These are the 3 positions. Ooh, I see. We are onto something. So it is in fact this position that's selected here. It's the second G that was reported in the VCF. So I think I'll just spoil it now because I think we have it here. So indeed, so we can see here that the stop codon reported was in this codon, but the variant or like these two variants right next to it actually also impact the same codon. So the way that variant effect predictor annotated this was it annotated these two variants separately. So it annotated just these two, declared the second one to be non-oncogenic, and then deleted it, right? So we are only seeing the first mutation. However, this is actually a deletion insertion. We should be taking them together according to the HGSVC guidelines. If there is one space between the variants and they affect the same amino acid, then we need to be merging them together. So yeah, we can see here that it's not actually a nonsense mutation. This extra one was supposedly rescued. Actually, in this case, that's a great point. This was not a reversion, like a rescue, and I can explain why. So like I said, the two variants separated should be described individually and not as a deletion insertion, except if they are affecting the same amino acid. So what did we do? Our team is called clinical genome interpretation. Since we have to present a clinically validated, like accredited report, we had to get our pipeline to re-annotate it. Oh, sorry. So what we did was instead of like, we went and directly edited the VCF field so that instead of just a G to T, it's actually like this deletion insertion. And we got, of course, predictably the variant that we hand pieced together. According to OncoKB, this one's actually unknown oncogenicity. So as a result, we actually removed it from the report. But we know that everybody likes to know about in interesting cases. So what we did was we informed the requisitioner, the person who asked for the sample to be analyzed. We let them know what we saw, but we just weren't allowed to put it on the report. So coming back to this data quickly, we can actually see that the allelic depth information, et cetera, like the total depth, et cetera, is almost identical or is identical. So that implies it's actually all one deletion insertion variant. We did have another case in which we had 3 different time points. The first time point in which was a nonsense, and the second time point which became TAT, which in that case it was exciting to see a reversion happen. Yeah, I think that's all from me. Yeah.
Thank you. Very interesting. It makes me very worried in that I need to check IGV every single time I do a report now from now on.
We always check them. I actually have like a whole bunch, like I have like 6, 7, 8 examples of when the calls were wrong and IGV revealed the real call.
Mm-hmm. How often would you say this happens? This type of, where IGV is calling the wrong variant and getting the nomenclature wrong. Does it happen in certain instances?
IGV. It would be like in our case we use variant effect predictor and other people use other like software. I would say it does not happen as often. Like in the 3 years that I've been working as a bioinformatician here, it's maybe happened like 10 times. So actually quite low, but we still check everything just in case. And we do find ourselves removing variants that could be artifacts, especially in targeted sequencing. Targeted sequencing, we're always finding artifacts at low VAF, like 1%, 2%. But in whole genome sequencing, we don't often see artifacts or weird calls. Relatively rarely, but not so rare that we would not check IGV. I don't feel safe not checking IGV.
Great. Let's see if there is a comment. The expression of the variant at RNA was 4%. Are there any other explanations for this allele expression imbalance?
In this case, are you talking about in the beginning? Where is it?
Oh, it's just an example of another report. Was not this one, right?
Oh, was it this one? No, the expression was 4%. Where is it?
I don't remember if it was only your example of a test or if it was the example of this specific variant. But anyway.
No, you're right. It was
the expression of the variant. Yeah. Okay.
Right. Yeah, we're not sure. So basically, like again, like what we do is we just present what we find in the RNA data. This expression 4% is actually relative to expression data of high-grade serous ovarian carcinoma. So this tells us that relative to ovarian cancer, this is like lower. Yeah, so it looked like it makes sense for a nonsense mutation for this to be lower. Yeah, we're not actually quite sure why. Like maybe potentially there's still a loss of function here, or I don't remember what the copy number data looked like, but there could be a copy number alteration in BRCA causing it.
Thank you.
Okay, so now we can move on to our last presenter. Jane Witt, are you here today? I do believe I saw you signing in.
Yes, let me share the screen.
Hello.
I am a little bit slow with Teams. Screen. Are you guys seeing the screen?
Yes.
Okay, so hello everyone. I'm Jane Witt Bongbunsen. I'm a Thai nephrologist. I was trained in Boston, still on service for the nephrology or kidney disease doctor, but I'm trying to set up a genetic program in Thailand, and we have our variant scientists, and I'm trying to learn as we speak because we don't have enough variant scientists in the country. So this very exciting case for me is what I receive after I see this girl who is 20 years old and unfortunately was found to have a very high creatinine, which is a measurement of kidney function at 2.0. For her age, this is very bad. This is like chronic kidney disease stage 4 on the verge of getting dialysis. So we would need to try to understand her disease. We get the kidney biopsy, and these are the findings. The light microscopy show a histological pattern of FSGS, which doesn't mean anything. It just means that the kidney has scarred out. There's no clues that we can get why this kidney scarred out from the immunofluorescence or IF or from the electron microscopy. There are some clues that there are immune complex depositing in this scar kidney. So from the clinical nephrology, all I can say is that this kidney has scarred out. Maybe it is an autoimmune process, but this by itself smells very genetics, because why would someone have a very silent autoimmune disease without any symptoms and then present it like this? So clinically, I can't tell what she had, and we do genetic testing, and this is the report that I get back for my genetic variant, which makes me puzzled because I can't find it on Franklin, and I think it's just coordination that are different. Eventually, the real variant that I can put it in Franklin or Warstrom is this one, so you guys can work from this one.
Jane Witt, did the testing company provide you with a reference sequence?
This is what they the NMID. Oh, I didn't copy paste the NM.
Okay.
Yeah, I didn't copy paste the NM, but eventually I tracked it, and it is this second line.
Gotcha. Just wanted to be sure. Thank you.
Okay, keep 1 more minute. I think I'm doing the timing for 2 to 3 minutes. All right. The detail of the report that I get back from this company is that it was signed out as a VUS. Cysteine are not too much different than tyrosine. It is absent in NOMAD, and there are several observations in patient with FSGS, which is the same histological pattern that I see in the patient. But FSGS is somewhat similar to like cardiomyopathy. It's just a scarred out kidney. It doesn't tell us much. If there are clues from the history, then I can favor this FSGS towards being genetic or non-genetics. But in her case, I would love it for it to be genetics, but it would start at the pre-test probability of around 30% being genetics. And the reported in silico predictors from this company said it is probably deleterious, and all I get is a VOS. So I go to Franklin is also VUS. Sad, I don't know what to do much. So I need to go back to the literature and try to look at all of these cases that are reported. And fortunately, these three cases are reported separately from three different ancestries. So they all come from Arabic ancestry from the US, and this scientific report is from Japan. Sorry, I missed the PubMed ID. And I know the first author of this scientific report came from Japan. So I did reach out to her to say, what are the phenotype of the patient? Because most of these patients are FSGS, and this one have asymptomatic proteinuria. It seems to be too mild. And what I find very interesting is that the variant scientist team from Japan actually wrote this back to me that for WT1, which can cause Wilms tumor, what we know is that in the exon 8 and 9, they are very important, but we are starting to learn that in exon 7, it is also quite important for DNA binding because it is a transcription factor. And the biology is that the cysteine residue here binds to this histidine residue. And these two duos are very important for the DNA binding. And the cysteine residue 398 is exactly at that location where biologically it should make sense. And in terms of the phenotype, I can't really get any phenotype to support this theoretical hotspot or functional studies, because what it is reported for the C2H2 sites in exon 8 or 9, it seems to be rather more early onset of the disease, like infancy or ESKD at early childhood. She's 20 years old. So I can't really apply what I learned from exon 8 or 9 C2H2 site. And then when it comes to other than exon 8 and 9, like what we have here, which is exon 7, it's variable. So I can't really use much clinical clue, but this is somewhat piqued my interest enough because there are three separate families in different countries. And then I went back to Boston, and fortunately, Boston children also have a children with this variant, but they haven't published. And interestingly, the patient at Boston children also have immune complex deposition, which is supposed to be the feature not suggestive of genetics, but they are also present in the same way that the immune complex are not bright, they're not in immunofluorescent, but they're just in the electron microscopy. So I'm more confident with the phenotype. And from what I reinterpret, I added the functional domain here of PM1. And then there are several cases that are rare enough in HGMD. Counting my case would be 5 sources, right? 3 in HGMD, 1 is mine and 1 in Boston children. And then in many of these families that are published, they segregate pretty well. These can be a little bit an overkill, but I need to decide something because these immune complex in the kidney can make the kidney doctor wanting to give immunosuppression to stop autoimmune disease, which can be accelerating the patient's kidney decline if we are giving them immunosuppression in someone who has already bad disease. So I need to give them some confidence that this is probably genetics, and then do the de novo. And fortunately, when I do the de novo, it is actually de novo. And that helps solve the case that this is probably likely pathogenic, which means that my decision to stop immunosuppression is a good decision, but it takes a lot of challenge and a lot of faith in myself to put this variant to likely path before the de novo result came back. That's all I had. Oh, did you lost my voice? Did I lost my voice to everyone?
No, Yasmin is
Oh,
on
okay.
mute, I think.
Oh, okay.
I'm sorry, I'm on mute. Thank you so much, Jane Witt. That was a very complex story, especially thinking about what I know about the WT1 gene already. I was expecting a story about Wilms tumor, and then we're hearing a story about an immune complex. So thank you for sharing that. I'm just quickly looking through OMIM to see, if I can see any associations. Are there any, I want to say, any well-known papers that you can share about WT1 and immune
Oh,
complexes?
yeah, that feature of immune complex are floating around, but we as a field haven't pretty much pointed it out. They're all very subtle at the range of non-specific immune complex, but in all other glomerulopathy, there shouldn't be immune complex if there are genetics. But WT1 would be the prime example of that they can.
Wow, that would be great to publish on once you sort it all out. Okay, so there's a comment here. WT1 has 24 different isoforms. I'm wondering which isoform this variant affected. Is this isoform highly expressed in kidney?
I don't know. How do I check that? Probably not just GTEx, I guess. That's why I need you guys.
Yeah, I was thinking GTEx. I don't know where else to look. Does anyone have any suggestions? GTEx would be a reasonable place to start. So apparently you can see it in Nomad. You'd have to play around in Nomad. That's not something I look at very often in Nomad. The PEXT track. Well, that's a good starting point. Does anyone else have any suggestions for Jane Witt, where he could go from here? Do y'all think long read would be good for that question? To know if, so would long read be good if the isoform is in the kidney?
Those are great suggestions. I'll play more in these tools to look into the exact isoforms and transcript.
Right. So you can always keep the chat available to yourself for when you want to work on this later. And with that, I think I will congratulate all of the speakers. Thank you so much for sharing all of this very interesting data. There is a lot to learn. I think what was so interesting is that we have people from all over the world, but also people in different roles doing variant science. So really, I'm just so pleased, and let's give everyone a big round of applause. So as I mentioned at the beginning, please fill out the feedback forms. It will help us with our next puzzling variant event in the future. Hopefully we'll have another one in the next few months. Thank you everyone for being here with us. Take care.